So this is going to be the first lecture of TBT 4165 that's done over YouTube. Let's see how this works. The topic for today will be knockout analysis. We're going to start by explaining reaction knockouts a bit more in detail compared to what we've done earlier, and both talk about single knockouts, which is what we've mostly talked about so far, and then generalize it into multiple knockouts, specifically double knockouts, triple knockouts, and so on. Won't really be discussed, but the concepts will be the same. And then we're going we're also going to look at the concept of gene knockouts and how we represent that in terms of reaction gene rules and the relationship between reaction gene rules, reaction knockouts, and gene knockouts. And lastly, we're going to tie in this concept of multiple knockout analysis into the biological concept of epistasis. So, to begin with, let us imagine the following model. It is pretty simple, and it is, of course, pretty unrealistic, but for the purposes of this example, it will do just fine. So, it's a pretty simple, straight pathway. We have two sequential reactions to begin with, then we have two alternate pathways that go in parallel, consisting of just like simple lines, each of them, and then they merge back together through this last reaction R8, forms the metabolite M8, and then from metabolite M8 we can produce the biomass through this reaction R11. So this is our metabolic map, and in order to have a model, of course, we need constraints. We're going to set R1 less than 10. All the other fluxes are internal fluxes, so they can be as high as they want. And of course, in practice, we would model that by saying that, for instance, R3 has to be less than 10,000 or something arbitrarily big. All the stoichiometric coefficients here are 1, and our objective function will be Z equals to the flux through R11. And what we find is that with the constraint R1 being less than 10, then the optimal value of R11 that we can get out is 10. That's pretty straightforward. We can have 10 flux through here, then 10 flux through here, because that's as much M1 as we have. And then here, we can sort of be balanced off. We can either have a flux of 10 going through the upper bound, or a flux of 10 going through the lower bound, or whatever values between those that would sum up to 10 would be also valid. And then we would have a flux of 10 through R8, and then that will lead to a flux of 10 through R11. So this is our basic model. It's pretty simple. Now, what happens if we want to delete some reactions from this? We're going to do some reaction knockouts. What happens if we block these individual reactions? Well, for instance, let's start by deleting R2. So deleting R2, that's typically done in the model by just setting a constraint of R2 equals to 0. And what we see here is that now that this link here is severed, we can't really produce any M2, and that of course means that neither R3 nor R4 can carry flux because we make no M2 to feed into these reactions, and that problem replicates downstream until we get here where we still can't produce anything, and of course then we can't produce any biomass. So the objective function, the maximum value of the objective that we can reach once we knock out R2 is 0. And since the organism can't grow with R2 deleted, we say that R2 is an essential reaction. So that is a single reaction knockout of reaction R2. Let's try another reaction. Here we're deleting R8. R8 is now deleted, and we see that that we have the same. R1 could in principle absorb, like take up metabolites, but then it would have to pass it all the way downstream, and here at M6 we wouldn't really be able to do anything with whatever flux we get in through R5 and R7, which means that de facto these fluxes are both blocked, and the fact that these fluxes are blocked will also block anything downstream of them. And in any case, we can't produce any M8. Because we can't produce any M8, then R11 can't have any positive flux, and so our objective Z, which is still R11, optimizing biomass, is 0. And so just as R2 over here, R8 is essential. Lastly, we can, another third deletion we can do is R11. Deleting R11 is the same as setting R11 to 0. And since our objective Z is still optimizing biomass, which is R11, our objective Z will also trivially be 0. So R11 is also essential. The last essential reaction, even though there's not like a particular image for that, is R1. Similarly, if we were to delete R1, trivially nothing downstream could be produced, and so the organism could not grow. So those would be our 4 essential reactions. So what happens now, for instance, if we delete something else? Well, if we delete R3, same as always, we set R3 equals 0 as a constraint. And with the constraint R1 now less than 10 as our constraint, we see that, okay, we cannot have any flux from through here. We can't make any M3. And so because we can't make any M3, we can't make create any M6 through reaction R5. But this lower pathway here is redundant. It provides backup for this upper pathway. And so just because this one is out, we can still, even though this one is out, we can still run a flux through R4 and R5, R4 and R7, use that to produce M6, from which we can produce M8, and from there on biomass. So what we see here is that even though we've deleted this reaction, we can still have a flux of 10 here, 10 through R2, 10 through R4, 10 through R6, 10 through R7, 10 through R8, and 10 through R11. So our objective function will still be 10, and that is the same as we had in the full model. That deletion of reaction R3 made no effect whatsoever on the potential growth rate of our organism. And because of that, we say that R3 is non-essential. So if we go back to our model here, what else do we have that is non-essential in this R3? Well, we have R4, R6, R7, and R5. These reactions are all also non-essential. If we were to delete these individually, we would still be able to have flow through the organism, because at most, knocking out, for instance, R4, that is going to knock out this entire lower pathway in its entirety, but we still have the upper pathway that is valid. Similarly, knocking out R5 is going to knock out the upper pathway just as knocking out R3 did, but the lower pathway is still valid. And in either case, since these are internal fluxes that we don't consider as being constrained, we're not going to have any form of reduction in what our biomass can be, because what's really constraining our biomass is the uptake rate we get through R1. And so as long as we can have internal fluxes going through, in this specific case, our objective Z will be 10. What happens now if we do a double deletion? Well, what we see here is that if we were to delete, for instance, R3 and R4, even though individually deleting them would make no impact, we see that deleting both of them at once, that will knock out both the upper and lower pathway. Because of this, we can neither produce M4 nor M3, and which are alternative precursors to M6. So M6 now cannot be produced under any from our model with R4 and R3 knocked out. And so because of that, R8 and R11 are also blocked, and so Z equals 0. R3 and R4 together are essential, and that's what we call synthetic lethality. They're examples of genes that are non-essential when removed in isolation, but when removed together, they will knock out the organism. Obviously, if we were to do something like deleting R2 and R3, that would also be knock out the organism. But any scenario in which a single deletion will knock out, will set the maximum growth to 0, any double deletion involving that single deletion will also trivially be 0. On the other hand, if we were to do another double knockout, say we delete R3 and R5, that is this entire upper pathway. Then, well, we still have our lower pathway that is valid, and our organism can still grow at this ideal rate of z equals 10. And because of that, R3 and R5 are not considered synthetic lethal. So, from a simple visual perspective, in the simplest case, the main scenarios, so to speak, that indicate synthetic lethality between genes has to do with whether they're parallel or sequential, and if they're in the same pathway. Sometimes reactions will be entirely independent from each other, and you might not see any effect. You would have no synthetic lethality. But in cases like this, essentially everything that is one after another will typically not be synthetic lethal, while things that are at parallel points along a given pathway will have a synthetic interaction. You might have, even then, something more complicated. We could have a third alternate parallel pathway here, in which case you would need a triple knockout to disable the organism's growth. But the principles are the same. So far, we've only really looked at, so far, the determination between whether something is essential or non-essential has been quite straightforward. If we look at this model, any deletion here is either going to knock out the model entirely and prevent it from growing at all, or it's going to have no effect whatsoever, and the organism is going to be able to grow at the ideal unmodified rate of z equals 10. So let's expand our little model to see. This is not always the case, so we'll expand our model a little bit and see the sort of problems we can actually run into in real life when we talk about determining whether or not a reaction is essential or not. So here we have an expand model. This top pathway here is the same as the one we had earlier, but we've added a very simple, just two-step pathway at the bottom here. R9 and R10. R9 imports metabolite 7 from the environment. R10 processes M7 into making M8, and then that can be then fed into the biomass. Since if we now say R1 less than 10 and R9 less than 10, well, we can produce 10 M8 through this lower pathway, and we can produce 10 M8 through this upper pathway. And since all of this M8 can be produced into biomass, our optimal value for the objective is 20. So that is the baseline we're looking at before we start making knockouts. Now, of course, if we were to knock out, say, R11 in this model, it would not grow at all, and then the model would be dead. And so R11 still remains clearly essential. But suppose now we delete R2. So we set the constraint R2 less than or R2 equals to 0. R9 and R1 are still less than 10. But we see that this upper pathway now, as earlier, cannot be used to produce M8. So we can only produce M8 from our lower pathway. And since that lower pathway is restricted by R9 less than 10, then we can only produce 10 units of M8 per time unit. And because of that, R11 can only reach a value of 10. And that is half of our original growth rate. And so the question is R2 an essential gene? And there is really no straightforward answer to this. That is a decision that when you run deletion analysis, you have to make a cutoff on whether you consider something essential or not. If something grows, if you make a knockout and you'll have a growth rate that's reduced by 3 to 4%, often you'll say that that is good enough. We don't consider this to be a lethal mutation, and so we consider that gene to be non-essential. Similarly, if something grows at a rate of 1,000th or 1,000,000th of the original growth rate, then you can say that it's as good as knocked out as well. But there are points in between. For instance, if your growth rate is a half or a third or something along that order of the wild-type growth rate, then you will have to make that decision yourself. Maybe you'll have you can include a third category that says, well, it's partially essential. That depends entirely on the analysis you want to run afterwards. And there is no straight, clear-cut answer. As long as you can justify with yourself for the purposes of your study what constitutes an acceptable threshold for considering a knockout to be lethal or not, that is fine. So from that, we can expand this. So we talked now about reaction knockouts, but reaction knockouts, controlling the bounds of reactions is not often that easy in real life. Typically, the only reactions where we can individually block a specific reaction are exchange reactions, and even then we can only block them partially. What we can do for exchange reactions is we can put our organism in an environment where the cell does not have access, isn't able to uptake that metabolite. And so then you would adjust the uptake rate and fix that to 0, which could effectively knock out that reaction if that is not a reaction that will plausibly secrete anything. But internal reactions cannot directly be constrained. What we can do is we can mutate genes. And by mutating genes, you might affect the reactions a cell can do, because in a cell, most internal reactions are dependent on certain gene products in order to be catalyzed. So we've talked about this earlier, and in any model we will typically have a table of what we call reaction gene rules. The reaction gene rules are essentially a set of rules for each reaction that specify which genes or combination of genes need to be functioning normally in order for their reaction to be able to take place. And this is done by a sort of Boolean binary syntax. So the way we consider is that every gene can be either active or inactive, and either reaction can be active or inactive. And so, for instance, we can have a very simple one-to-one relationship, as we have here, where reaction hex 1, in order to be able to flow, requires that gene B23A8 is active and unperturbed. And that is it's a pretty common case, and is also very simple. But we can have more complicated cases than this. We can, for instance, have isozymes, where we have two alternate gene products, that either of which are sufficient to catalyze the reaction. So in order for G and K to be able to flow in this case, we just need either B4268 or B3437 to be able to produce gene product. And so that is represented by an A or B logical rule. The third case is where we have a protein complex where essentially two separate gene products combine, and that combined gene product is necessary for the reaction to flow. In that case, as we see here, for A and S to flow, merely B1264 alone or B1263 alone is not sufficient. We would need both of them to be present. That is equivalent to an AND check in terms of Boolean logic. And so these are our two main rules that we have. We have AND or relations, and as a third interesting aspect, a single gene can have relevance to multiple reactions. For instance, here we see that the gene 3916 both individually governs reaction PFK2, but it will also serve as an isozyme to reaction PFK, which means that in this case, if we were to knock out B1723, reaction PFK could still run because B3916 is still available, and reaction PFK2 could still run because PFK2 does not depend on B1723. If we were to knock out B3916, PFK would still run if B1723 is available, but PFK2 would be knocked out. So it's not a one-to-one relationship. Any given reaction can be governed by multiple genes, whether by or an AND relations, or an individual gene can have an influence on multiple reaction. The last thing that can make this sort of complicated at times is that these reactions can be nested. For instance, you can have scenarios where a protein complex acts as an isozyme for a reaction. So we could, for instance, have a reaction where we either need B3437, but if B3 or the combination of B1264 and B1263. So none of these would individually be necessary, but 3437 would be sufficient alone. B1264 would not be sufficient alone because it needs B1263 to work as well. So what happens if we put this into our model? If we put our gene rules into our model and run that? Well, we can note something like this. So if we want, I will let you now pause if necessary and try yourself to see what happens for each gene deletion. If you delete G1, that will knock out R1. What effect will that have on our metabolic system? If you knock out G2, what effect will that have in terms of reactions? Which reactions does G2 participate in? And what effect would knocking out those reactions according to the specified rules have on our system? And similarly for each individual gene. The notation here is the vertical bar corresponds to an OR relationship, so R2 is requires either G2 or G3 to be available. So knocking out just one of them would not knock out G2 or G3. While R3 is dependent on both G4 and G5 being present. So knocking out either G4 or knocking out G5 would be sufficient to knock out G3. So I will let you pause and try to figure this out for yourself. If not, we can continue. Okay, so let us go through this one by one. Knocking out, if we look at G1 first, knocking out G1, we're still assuming that R1 and R9 have this original constraint that each of these are less than 10. G1 only figures in R1. So knocking out G1 would knock out R1, which, as we've seen earlier, would knock out this entire upper pathway, meaning that we can only produce M8 through the lower pathway, which, as we've seen before, corresponds to an optimal growth of 10, which is half of our wild type growth. If we were to look at G2, G2 only figures in reaction R2, so knocking out G2 alone, as we've said before, is not sufficient to knock out reaction R2. And so our pathway will be the same. We will have no change in available reactions from knocking out the gene G2. And by extension, we will still reach that optimal wild type growth where R11 is both fed by the upper and lower pathways and grows at a rate of 20. G3, as G2, only figures in reaction 2. And so it will necessarily, deletion of G3 individually will have the same effect as an individual deletion of G2. So the G3 similarly will correspond to a growth rate of 20 if you delete it. Now it gets a bit more interesting. Now let us move to G4. We see that G4 figures in 3 different reactions. We see that it figures in R3, in R4, and R9. We can start with the simplest one, by R4. We see that G4 singularly governs R4. So knocking out G4 will knock out R4. That will knock out our entire lower pathway here. Similarly, G4 for R3, knocking out G4 will knock out the complex. This and sign means that both G4 and G5 need to be present. So knocking out G4 will also be sufficient to shut down R3, and that will shut down this upper pathway. Now both this lower pathway and upper pathway here have been knocked out. And that means that this entire upper half, this entire extended pathway that converts R1 into, sorry, M1, it takes in M1 and converts it into M8, is blocked. So if we were to have any growth, we would need to get that from our lower section. But as we see here, in R9, G4 and is part of a protein complex with G7 and or G8. What that means is that knocking out G4 is also enough to knock out R9. And that, as we can clearly see, is enough to knock out this entire lower pathway as well. So we see that by knocking out G4, we have knocked out this, and this reaction, R3, R4, and R9. And with all of these three cut off separately, we cannot have any growth. And so our optimal Z for the G4 mutant is a growth of 0. If we look at G5, G5 only figures in reaction 3, but it figures as an AND. So knocking out G5 will knock out reaction 3, but reaction 3 in itself is not critical. And because of that, we will still reach full optimal growth even when G5 equals 20. Even when G5 is knocked out with a growth Z equals 20. Moving on to next gene, G6, we see that G6 directly governs reactions R5 and R7. So knocking out will knock out both of these reactions. As we see here, that is sufficient to knock out the entire upper pathway. And so we can now, again, only resort to the bottom pathway. And so our rate of R11, our production of biomass, will be restricted to whatever amount of R9 we can take in, which is 10. Moving on to that, G7. Knocking out G7, I have actually made a mistake here in the slide. We see that knocking out G7 will not have an effect on the on R9, because if G8 is still available, then G7 is not necessary. As long as G4 and G8, for instance, are available, R9 can flow. And since G7 is not present in any of the other reactions, the single reaction mutant, the single gene mutant G7 will not have any different capabilities in terms of reactions, and so will grow at an optimal rate, as will G8. So in this row here, the values for Z optimum should actually be 20. Looking at our last candidate gene is G9. G9, as we see, only figures in reaction 10. Knocking out G9 would then therefore knock out reaction 10, which bars our entire lower pathway, but we have access to this upper half, which as we've seen before, can produce biomass at a rate of 10. So here we have our table with the various optimal growth rates for the various single gene mutants. And what does that tell us about the essentiality of these individual genes? Well, that depends on our criteria, as we've said before. For G2, as for G7 and G8, despite the fact that these are mislabeled, and for G3, the single mutant will grow with full fitness. And because of that, the gene is unambiguously non-essential. Meanwhile, for gene 4, we see that the deletion of gene 4 does prevent entirely the growth of the organism. And so that gene is clearly essential. Meanwhile, the remainder genes, G1, G6, which corresponds to this, and G9, their mutants will grow at a reduced rate. And whether or not that is considered a valid or a sufficient reduction in growth rate to consider the knockout as lethal is a matter of judgment for that particular analysis. So this is a basic overview of how we would do single gene deletions analysis on a given organism. So in practice, how this would be done in COBRA would be that you tell the program which genes to delete. It will readjust the model with the reactions blocked according to the genes you've deleted, and then that will become a new FBA problem that we'll try to optimize, and it will return to you a growth rate, and that growth rate that you from that single FBA analysis will tell you the fitness of that mutant. But that is single deletions. We can expand on this a little bit, because there's a very important concept when we're talking about genotypes and phenotypes called epistasis. Epistasis refers to conditions where the act or the activity of one gene product either accentuates or masks the effect of another. So as an example here, we can look at different phenotypes from mice. So here we have 2 different genes with various alleles possible, either uppercase or lowercase. And what we see is that we can sort of consider the black to be the base phenotype. We see that if both of the B genes are in the have the lowercase allele, then we will get a brown phenotype. So we will not, merely one allele switched will not be sufficient, but adding a second one will suddenly have a dramatic change. And that is an example of what we call aggravating epistasis, where one modification has no effect. And the second modification also would have no effect in isolation, but combined they do have an effect. For the albino genotype, well, we see that is governed by this C gene here, and we see, as with the brown, one C, lowercase c, is not sufficient for the albino phenotype, but 2 is sufficient, and that is similarly a case of aggravating epistasis. But what we also see is that for the case of both the B gene and C gene being in the relevant allele for both our brown and albino genotypes, we will get an albino phenotype. What this means is that these C genes here, when they are present, they will mask whatever happens at B. And this is something that happens quite commonly in biology, and that we want to be able to identify. Now, you can have one mutation that becomes invisible simply because there is another mutation that overrides it in a certain way. And so if we're looking at this coloring here for mice, it's a nice original example, but it's very certainly a relevant case of epistasis. But of course, we can't tell the color of an organism based off FBA. That is not really part of what we do. If we want to have a bit of a more formal approach to defining epistasis, we want to have something that we can quantify. And quantifying the color of a mouse is not necessarily something that you can put a good number on. Instead, what we can do is we can look, for instance, at the fitness of an organism as a numerical value. For us, that would typically be the growth rate. That is a good example of what could be considered as the fitness of whatever organism we're modeling. And we could see what happens when we mutate, as we add mutations. We might do several mutations in a row, and the first few mutations will not have a particular effect. But after you add a certain amount of mutations, then your fitness starts dropping off quite dramatically. And that is a case of aggravating epistasis, where where the sum of multiple mutations will have a much more powerful impact than what they would just do individually, added together. Meanwhile, the opposite case is what we call buffering epistasis. In buffering epistasis, we will have a very marked effect by one or a few mutations. And after that, if we add more mutations on top of it, those mutations might not be particularly remarkable in terms of their effect. Why do we care about epistasis? Well, it's because they are quite difficult to figure out. There's a lot going on with epistasis, and it's quite hard to measure. So, in terms of figuring out what we don't know about an organism, epistasis will often be a good source of information because it's something, if we can find out what governs epistatic interactions in our organism, we're stepping into uncharted territory, and that can often be quite useful. So how does epistasis relate to the concept of multiple knockouts, or to metabolic pathways? Well, here's a reasonably simple map of a metabolic pathway. And we see that epistasis quite closely relates to this notion of multiple knockouts that we talked about earlier, where we have either parallel or sequential pathways. So for instance, if we were to knock out this reaction here, that would not have much of an effect because we have redundant pathways available. Meanwhile, if we were to knock out this one after that, we would still see no effect on no added effect because when this is knocked out, this reaction is already constrained. Deleting the gene that governs this reaction will not constrain it any further than the implicit constraint that is imposed on it by the deletion of the downstream reaction. Meanwhile, if we were to delete both this and this reaction, now we might have an actual loss in fitness, because if this upper pathway is less efficient than the lower one, knocking out this bottommost reaction in addition to this one would be enough to knock out the entire lower pathway. And so a single mutation might not be sufficient to see it, but a double mutation would have a pretty dramatic effect. And that is an example of what we call an aggravating epistatic interaction. So how would this look in our model? Well, what we could do is we can do every possible combination of double mutants and see the growth of our how this pathway would be able to carry flux and how our organism would be able to grow. So we can, for instance, look at what happens if we delete both G1 and G2. Well, what we will see clearly is that deleting G1 and first of all, deleting G2 will have no effect on its own. Deleting G1, on the other hand, will have some effect on our total fitness, but since deleting G2 has no effect that we can see, it is a neutral epistatic interaction. Essentially, since this is a full fitness, the G2 mutant is full fitness, and the G1 mutant is partial fitness, and the G1 plus G2 double mutant has the same fitness as G1, this is a neutral epistatic interaction. Meanwhile, if we were to look at, for instance, G1 and G9, we would have, for instance, let's compare rather G2 and G3. If we were to look at the G2, G3 double mutant, it would have a dramatically reduced growth because the G2, G3 double mutant would not be able to carry flux through any of this upper half of the pathway, or of our model. While the individual mutants G2 and G3 each have full fitness. Any double mutant that involves a mutation in G4 would have already zero fitness, since any single mutant in G4 is unable to grow. And we cannot increase the maximum growth by adding more deletions. So the notion of epistasis for essential genes in this context is not particularly interesting. Meanwhile, if we were to compare, for instance, G9 and G6, they would be similar to G2 and G3 in terms of epistatic interactions. No, never mind. G9, I thought this was G9. No, G9 and G6 would each have a partial phenotypic effect on their own. Each of the, like any single mutant of either G6 or G9 will have half the fitness. Meanwhile, the double mutant of G6 and G9 would have 0 growth. And that is an example of an aggravating epistatic interaction as well, because we consider epistasis to be governed by the product of the fitness of each mutant. As we can see here, the way we measure epistasis is we take the fitness or the growth of the double mutant, subtract it from the product of the fitness of each of the single mutants. So for instance, if the single mutants each reduce fitness by half, the product of this would be 1/4. That would be a no epistatic neutral epistatic interaction, would be that the double mutant had half of the original fitness, which means 1/4. So if we have an epistatic or a double mutant of 2 half fitness genes where we end up with 0 fitness, that is still an aggravating epistatic interaction. Of course, if both the single mutants have full fitness and the double mutant also has full fitness, that is a non-epistatic interaction. And similar, if both of them are lethal individually, of course, the double mutant will be lethal, so that is also a non-epistatic interaction. And so we can use FBA to identify epistasis by running tests for all possible double knockouts of our model and see what their resulting optimal growth rates are. And we can classify and by comparing the double mutants to the relevant single mutants, we can classify epistatic interactions. This is typically only done for double knockouts, because FBA takes some time to run. So a single FBA solution feels quite instant. It will be something along the lines of a fraction, like a smaller fraction of a second, depending on your computer, everything, and your model, anything from a millisecond to maybe a full second. A single gene knockout scan involves making one mutant for every gene and then solving that as an FBA problem. And in this case here, if we have 10 genes, that means we have to solve the FBA problem 10 times. For a real model, we're often looking at something more along the lines of running 1,000 FBA simulations, which, if you have something that is 1,000 times something that takes 1/10 of a second, that takes 10 seconds, which is still reasonably fast and accessible. If you want to do a double gene knockout, well, suddenly now you have to test up to the number of genes squared. Certainly you can cut out all the essential genes from your second knockout. So you might have, for instance, so if we were to run a double knockout here, we would first have to run 9 single mutants, because that's the number of genes we have. We will find that G4 is essential, and so we can discard that on the second round. But that still gives us 8 genes left, and all combinations of these 8 genes is 8 multiplied by 7, which is 56, divided by 2, which is 28 double mutants. For a real model, when you're talking 1,000 genes, you might have maybe 200 of them that are essential on the first run. And that means that on the second run, you would have do something along the lines of 600 or 300,000, 400,000 double mutants, which takes a lot more time than simply running one FBA solution, right? It takes 600,000 times as long. And of course, if you can run it, if one run takes 1/10 of a second, 64,000 seconds, that is the better part of a day. And this will scale exponentially, or almost exponentially, with the number of knockouts you want to run, unless you're making qualified guesses or have other ways to restrict your if you have other ways to restrict the number of or the actual set of genes that you're trying to knock out. Of course, if you're only looking at 10-20 genes of interest, you might be able to do quadruple, quintuple knockouts without having to do a prohibitively large amount of FBA runs. So how does this look in an actual model? Well, we'll see here that if we look at epistatic interactions between genes, different genes annotated for different processes, we see that there's a strong tendency for genes in the same functional group to either be fully aggravatingly epistatic or dominantly buffering epistatic. And that will often quantify how redundantly and how robust that pathway is, or sorry, of that process is. So for instance, if you look here at mitochondrial membrane transport, it will typically have, like with itself, and for that matter with other genes as well, we'll see often a lot of buffering epistatic interactions indicated by these green circles, which indicates that multiple deletions are not particularly aggravating in that specific pathway. While these red dots here correspond to pathways that are quite vulnerable to multiple deletions. Well, we also see that epistatic interactions can happen at quite remote and quite complicated and not necessarily intuitive ways. If we were to compare to our original model here, we can almost read from sight what the epistatic interactions will be. In a real model, where pathways are larger and more complex than the epistatic interactions might not be clear from visual inspection. And you're actually dependent on simply running the FBA simulations and seeing how the cell will respond to multiple knockouts compared to these individual knockouts in isolation. And yeah, that I think sums it up for today. I'm not quite sure how long this got. I think it's a little bit shorter than a normal class, but at least you'll have the opportunity to return to any point that you want in case I'm going too fast, and just look over those parts again. Thank you.